Okay, so I can't see what's on the screen. Can you see what's on the screen? Okay, good. So hello to all of you. My name is UP Bhalla. I am a scientist at the National Center for Biological Sciences, NCBS, which is a daughter institute of the Tata Institute of Fundamental Research. Now, I thought they asked us to give a little bit of an introduction to ourselves. So I don't know, how many of you have done physics? A lot of you have done physics. How many of you have done biology? The same hands came up. So at least when I was in college, the general idea was that some people would do physics and some people would do biology, and they'd never be the same people. How many of you are planning to do just physics or just biology? Yeah? So the thing is that what I'll be talking to you about comes about, it's possible because I have done both physics and biology in my career. And this idea of interdisciplinarity, that's a long word. You've heard it, right? Interdisciplinarity? Yeah? It means that you bring the strengths of many fields of research to understand really difficult, complicated, and very interesting problems. So that is what I have done throughout my career. I've used some physics background, a lot of biology background. I put them together, and it allows us to study what I think is the most wonderful system to study, that is the brain. So that's what I'm going to tell you about. Specifically, I will talk about memory. And so let's get started. So the brain is not, like a single magic black box in which all of these things happen. The brain actually is composed, as you might imagine, out of molecules and atoms. They organize into proteins, those organize into cells, and cells are organized into circuits. And the computation of the brain cannot be exclusively imagined to be at just one level or just another level. The computation that the brain does is very much a case of signals traveling up and down between the molecules, between the genes, between the circuits, between the cells. And it is from this interaction between levels that brain computation happens. So what I'm going to try and do is to give you a glimpse of how brain memory happens, again, across many, many levels. I will start, as you'll see, with molecules. So let's just look at some sizes, right? So this is the size, this is a brain, right? Your brain, my brain is about 1.4 liters, 1.5 kilos. And you can see, at least on your screens, and I don't have a screen here to see, you can see roughly how big is it. It's that big, right? It's that big. Okay, so now if you look very closely, can you see a little spot to the right of the screen next to where it says size of a neuron? Yeah? So in that huge brain, your brain cells are about 0.5 millimeter in size. Because of their branching, the cell bodies are much smaller. The branching structure of a brain cell is about 0.5 millimeter, and you can see it there in the corner, right? Yeah? Okay, so now what we're going to do is we're going to zoom into a brain cell. Okay, so this is one of the brain cells. This is called a Purkinje neuron. It's an absolutely beautiful brain cell. This is actually a computer model of a brain cell, but it's a close representation. And you can see all its branching, all its dendrites. These are called dendrites, those little branching structures. And now, can you see in the corner of the screen another tiny little dot which says size of neuropil? Yeah? Okay, so now what we're going to do is we're going to zoom in and focus on the neuropil. What is the neuropil? The neuropil is just the nervous tissue in a certain volume. Yeah? So what we're going to do is we're going to zoom in to a size of 5 microns. And let's look at the neuropil in 5 microns. Okay, so now this little cube of tissue has got exactly one dendrite. That's the yellow sort of central tree-like structure going through it. And it's got all sorts of branches and connections and so on around it. Yeah? And if you look very closely, you can see a lot of little red spots. Do you see the red spots? Those are contact points between this cell, this dendrite, and many, many other inputs coming into it. Those are called, what are they called? They're called synapses. Very good. Yeah? So those synapses are indicated in red, and you can see that tiny little dot on the corner which says size of a synapse. Yeah? Okay, so now we're going to zoom into a synapse. Okay, so this thing is the synapse. It's about half a micron across. In fact, this is just one side of a synapse. All right? So we've been zooming in, zooming in, zooming in. Now we are so small. This is half a micron. Can you see the little granule-like little blobs all over the synapse? And you can see some little football-like things. So the little granule, the little dots are proteins. And the little spheres are vesicles. They are the packets which contain the neurotransmitter. Okay, so the point of this exercise was to give you an idea of scales, right? We started with the scale of a head, and then we zoomed into the scale of a cell. Then we zoomed in still further to the scale of the branches of a cell, and now we've zoomed into the scale of the synapse. And the reason I've done this zooming in is because brain function is happening at all of these scales. And what we are going to be doing in the next few minutes is to be looking at the computations that can be done at the scale of molecules. Okay, but let me just give you some numbers. Okay, so in your brain, you have roughly 10 to the power of 11 neurons. Okay, the current accepted value is something like 85 into 10 to the power of 10. So it's close to 10 to the power of 11 neurons. That's a lot of cells, right? But every single cell in your brain makes contacts, makes these synapses with a huge number of other cells of the order of 10,000 other cells. So you multiply 10 to the 11 by 10 to the 4, and what do you get? You get 10 to the 15, roughly 10 to the 15 synapses. Okay, now 10 to the 15 used to be, when I was your age, it used to be a really big number. Now it's a smaller number. Why is it smaller? Well, when I was studying neuroscience earlier, I'm still studying neuroscience, but when I was your age, the idea of storing 10 to the 15 bits of information was completely insane. Yeah? You could never do it. Okay, now tell me, how many bits in a gigabyte? Anybody? That's 10 to the 9. Yeah? What's the next one up? After gigabyte, you have terabyte. How many bits in a terabyte? 10 to the 12. Yeah? What's the next one up? Petabyte, very good. How many bits in a petabyte? 10 to the 15. Believe it or not, we have in our institute, some of you have in your institutes, machines which have more memory capacity than that. Okay, so the number of contacts in your brain is huge, but astonishingly, due to modern technology, we now have machines in the basement which have that same capacity, at least in terms of storage. All right, so as I said, these used to be really big numbers, but now, yeah, they're within people's reach. Okay, so no matter what technology says, you heard the previous lecture, right? The capacity to do computation increases to amazing degrees when you have this kind of processing capacity. Here we're talking about 10 to the 15 or so bits of storage. That is an incredibly huge number. Yeah? You probably don't ever use it. I mean, I'm sure you wish you could store 10 to the 15 bits of memory when you wanted to write your exams, right? Yeah, well, okay. Memory works in a much more interesting and subtle way, and I'll try and give you a glimpse of that. Okay, but let's keep going down this, right? What are the time scales of things that happen in your brain? Okay, so how many of you know how fast you can respond to a starter's gun in a race? Yeah, so if you're an ordinary human being, you can do it maybe in 0.3, 0.4 seconds. If you're an Olympic level athlete, you can maybe do it in 0.25, 0.2 seconds. Yeah? So that is 100 of the order of 200, 300 milliseconds. But, you know, to respond to the starter's gun means that sound has to reach your ears. It has to be converted from sound energy to electrical signals. The electrical signals now have to go through the brain, to the hearing part of the brain, then it has to go to the processing part of the brain that says, okay, what am I supposed to do when I hear this bang? And then it has to go from there to the neurons that say, okay, now I have to start movement. And it has to process which muscles to pull and all that, and then the signal has to go down to your muscles. Okay, all of this is happening in of the order of 100, 200 milliseconds, right? So it turns out that the fastest signals that happen in your brain are of the order of 1 millisecond. There are faster events in the brain, but the signal flow, the fastest that you can get is of the order of 1 millisecond, which is pretty jolly fast. Okay, but now let's start to take these time scales and explore them as they go down the list, right? So the electrical events are of the order of a millisecond. Chemical events are of the order of 100 milliseconds. Though, mind you, the neurotransmission, believe it or not, is mostly based on chemistry. Electricity is a sort of secondary part of it. So even chemical signals are extraordinarily fast. So now you go down the list. As you can see here, chemical events are of the order of a second to 1,000 seconds. You can have structural events, that is changing the connections, changing the shape of cells. That can be hundreds of seconds to months. And now we get to a very, very interesting number, which is what is the lifetime of a protein in your brain, or of any molecule in the brain? And the answer is it's of the order of minutes to days. And that takes us to the next point, which is what is the lifetime of a memory? Okay, so how many of you see that there's a very interesting paradox here? Yeah? That the molecules that make up your brain have a much, much shorter lifetime than the memories. And of course, the lifetime of the cells in your brain are 100 years. You don't, except for a very small amount, you don't have neural regeneration. That doesn't happen typically. So, in order to store information, you're using molecules, and yet those molecules do not last very long. So here's an essential paradox, and I will try to explore that with you. Okay, so how many of you recognize this experiment? Yeah? Pavlov's experiment, yeah? So actually, you know what Pavlov was really studying? He wasn't studying memory. He was studying digestion. He was interested in the generation of salivary fluid, and then he was going to use it to do enzymatic assays. But being a very good scientist, he observed something. This is called serendipity. He made an unexpected observation, and he followed it up. And what was his unexpected observation? Yeah, what was it? It was that when the dog sees the food, it salivates, right? Everybody, that's not unexpected. That is expected. When the dog hears a bell, it says, huh? Yeah? What has the bell got to do with it? But what was happening was that in his lab, every time the food was to be given, just for the instruction of all of the caregivers of the dogs, the bell used to ring, and then everyone, I bet if we took a recording from many of your salivary glands and rang the school bell, yeah, you would get salivation, would you not? Right? So there, you've been conditioned, Pavlovian conditioning. So that's what happened to the dogs, yeah? The bell came, and then the food came, and this association happened again and again and again and again. All right? And finally, the dogs are conditioned so that when they hear the bell, they salivate. All right, so now what I'm going to do is I'm going to take you to a very, very simple dog. This is a 3-neuron dog. Most dogs have a little bit more than 3 neurons, yeah? A little bit more. In fact, even flies have a lot more than 3 neurons. But let's take this 3-neuron dog, right? So the middle neuron is the one that responds to food, right? So when the food comes, the neuron, everybody knows what a neuron is, right? It's a brain cell. So when the food comes, this cell becomes active, and that causes salivation. Straightforward? Okay, good. Now, if randomly a bell goes off, it's loosely connected to this neuron, but not strongly. So nothing much happens, right? But now, over many, many repeats, what happens here, and let me see if I can indicate with a pointer. Okay, it won't let me. What happens is that the synapse, that is the connection from the bell neuron to the food neuron, because it's repeated many times, that connection gets stronger. And when it gets stronger, what happens eventually is that when you trigger the bell neuron, that connection is strong enough to then trigger the food neuron. And so then in this magical 3-neuron dog, you are able to learn Pavlovian conditioning. That is, you give the bell, and the animal salivates. Okay, so this is an example of conditioning, and we've sort of simplified it to 3 neurons, but the general principle is actually, in many cases, quite similar to this. That is, you have an association between the input and output cell. You repeat that association often enough, and you end up with a learning that happens because the connection between the 2 cells has been strengthened by repeated association. And this is a good way to establish learning, and many kinds of learning happen in this manner. Okay, and you can have another stimulus, let's say light, and it can go on just as many times, but if it is not associated, then the synapse does not get stronger and the animal doesn't learn. Okay, so this is sort of circuit level description of what might be going on when you learn Pavlovian conditioning. Now, I'm going to take this a little bit deeper, right? The question is, fine, I have said that the connection, the synapse, becomes stronger. I'm going to now explore the question of what could happen inside this synapse, this incredibly complicated bag of proteins. What could happen in there to store the information, right? It's not enough to say that there's an association. Something is happening, something is changing in this synapse, and it is changing for a long time, maybe 100 years. How do you store this information? Okay, so let me just make a point which perhaps is obvious, maybe it isn't. And that is that you're all very familiar with the idea of representing information as electricity, right? You use electrical signals for your mobile, you use them for telephones, you use them for all sorts of things. But you can just as well convey information through the use of chemistry. So a high value of a certain compound could mean the presence of a signal, a low value could mean its absence. And because there are thousands of chemicals, you can have thousands of signals represented in parallel. And we won't get into space just yet, but you can imagine that different domains of the cell may have different signals going on. So information can be represented as chemistry. So now let's get into this a little bit. Here's an example of someone. Do you know this guy? Yeah, did you see the movie? Some of you should see it. It's a nice movie. It's a nice movie. It doesn't capture all of it. This guy was an unbelievable genius. You know, he did code breaking, but that was the least of it. I mean, he helped win the World War II, but that's minor compared to the fact that he established the principles of computation, the fundamental principles of computation. The next lecture theater is named after him, the Turing Theater. Yeah? He also established many aspects of information theory, along with Shannon. And he also did this on the side. He discovered how you could use chemical signals to encode space, right? So all these patterns that you see, things that look like leopard spots and stripes and so on, these can be implemented in chemistry. Just another example of chemical information. In fact, here's a little movie. I don't know if it's playing. Can you see the twirly patterns, right? So if you stare at them too long, you'll get hypnotized. So don't look at them too long. Yeah? So anyway, these patterns are Turing patterns. They're patterns in space and time, and they result from chemical interactions. So this is one kind of chemistry. Let me go to another kind. So here's the synapse. Remember this horrible slide? Yeah? Incredible number of proteins in there. Okay. Here is a model that I made some 20 years ago with maybe 0.1% of the different molecular species in there. And this is a complicated model. This is what it looked like on my computer in the good old days. Yeah? It was horrible. You can imagine how much work it took me, let alone the people who provided the data. Okay, so this is a model. But we are used to memory of this kind. What is this kind of memory? This is a hard disk. Yeah? Oddly enough, this is becoming obsolete. What do you have now? You have SSDs, right? Solid state disks. Anyway, this is the hard disk. Now, this stores information in a particular way. What's the trick for this storage of information? It's magnetism, right? You apply a magnetic field and the hard disk retains that magnetic change in lots of little spots. Okay? So now this is what I call persistent stability. Once you apply the magnetic field, the signal stays where it is and it retains that for a long time. And you can do something like that if you induce structural change in synapses. Here's another kind of memory. What kind of memory is this? This is RAM. Very good. Someone is keeping an eye on it. Yes. This is RAM. And in this kind of memory, what you need to do, you need to keep refreshing the memory. You need to keep recirculating the information, otherwise it gets lost. And there's an electrical equivalent, a neuron equivalent of that, which I've indicated underneath, which says that if you have a little ring of neurons connected to each other, you can have reverberatory activity. You can have activity going around in a circle. And it's hypothesized that something like this happens when you briefly remember a phone number. Yeah? You have to keep saying it, and it'll go away very soon, but you can hold it for a little while. Yeah? So something like this, reverberatory activity. So this is another form of memory at the circuit level. But now, if we want to store information for a really long time, we use something like this. How many of you recognize this circuit? Oh, very good. So these are people who have studied computer engineering or Boolean gates or something like that, right? You did that in school? Maybe, maybe not yet. Anyway, this is a little circuit called a bistable switch made of NAND gates. And what it does is that it stores information as long as power is applied. And its crucial element is this feedback loop. And we're going to talk about feedback loops because chemical feedback loops are very, very common in the brain. So let's look at okay, oh yes, this is a painting, a very famous painting. Before I get into feedback loops, what is the name of this painting? Anybody know? This is Salvador Dali's painting by the name of Persistence of Memory. That's why I use it for all my talks, right? And the reason I use it is that we were talking about the synapse. Yeah, so this is my poor drawing of a synapse, this little mushroom-shaped thing. The reason I draw this is that a synapse is a terrible place to store information. Why is it terrible? It's terrible because, A, as we already discussed, the molecules will turn over, they will die, but the memories have to stay. It's also terrible because it's so small that there's a lot of chemical noise. I won't go into that. And there's a lot of diffusion of molecules. I won't go into that. So I'm going to ask the question, how do you store information even though the molecules turn over? And the answer is through feedback. Through these feedback circuits, you can store information. So let's suppose you have a molecule A and a molecule B, and you can see the feedback, right? A activates B, B activates A. Right, so now supposing you trigger strong activity in this circuit. Now you get a state where A is active, therefore it's turning on B, and B is active, therefore it's turning on A. Everybody got that? So in principle, you can store information. What does this have to do with molecules? So now let's take this down to the molecules. Those are the little circles and triangles underneath. Here's a bunch of molecules which have to store information. And what we're going to do is we're going to imagine that these two states, high activity and low activity, are like two energy wells. They're like two wells. The marble can either be in one well or in the other well. If it's highly active, it's in the, say, right-hand well, or it's in the left-hand well. So now apply this analogy to a bunch of synapses. Yeah, so here we have those molecules. They're active, they're turning each other on. And now what happens over time is some of the molecules die. That's the crosses. They die, they go away. If this process were to continue, you wouldn't have any memory left. But your cell is alive, therefore it keeps making new molecules. New proteins come in. And because there's already a preponderance of proteins there which are in the active state, say, the new molecules come in and they become activated. And over time, all of the molecules can die, and yet the memory is retained. Yeah? This is like, say, oral traditions in Indian scripture, right? You had people memorize the scriptures, not necessarily write it down. They would pass it down. The guru would teach his shishyas and so on over many generations, his or her shishyas. And then over generations and generations, the teachers would die, but the information would remain. And that is essentially what we propose can happen. So this is an answer to the paradox. How can you remember things even though the proteins that make the memory are dying? Okay, and so this, just to remind you, then this is what we suggest. Something like this, some kind of chemical feedback is taking place in those synapses which have been strengthened in order to retain the information for a long time. Okay, so now I'm going to go back and take you up many levels of computation to the brain as a whole. Yeah? And I'm going to introduce you to this gentleman, Henry Molaison. He is very famous His name only became known recently when he died, because he was, in the literature, he was called patient HM. And he is very important because his study of his affliction helped to answer the key question of where is information stored in the brain. It helped. I'm not saying it gave the full answer, but it helped. So Henry Molaison was unfortunate in that he had extremely severe epilepsy. And the doctors tried everything, and eventually they decided to do surgery and remove the focus of epilepsy. And what they removed was a part of the brain called the hippocampus. And if you see the diagram there, you can see the large holes in his brain. Yeah? You see these big holes? So that is what the surgeons took out. And as a result, Henry Molaison had memory deficits. He had extremely severe memory deficits, and I will explain what kind. He could not remember anything new. So in other words, if you introduced yourself to him, he would store it in working memory. So he would have a conversation with you, but if you step out of the room, 3 minutes later, come back, he's forgotten. But his long-term memory, what had happened before the surgery, that was perfectly intact. He could tell you the name of the president of that time. He could tell you I don't know, whichever team was winning in baseball, this is America. He could tell you all sorts of things from before his surgery. And of course, as he grew old, this was actually very tragic, right? Here is a man who's getting old. He knows he's getting old, but his memories are all of him being as a young man. Okay, so Henry Molaison, the analysis that they did, Brenda Milner and I'm blanking, Scoville and Milner, they did this. Over many years, they studied patient HM, and they discovered that, surprisingly, he was able to learn some things. He was given a task to trace out some shape. And that takes a little bit of practice. And the funny thing is that although he couldn't remember having ever done the task, he grew better at it. Okay, so what we learn is that there are different kinds of memory, and one particular kind of memory, the short-term memory, which then eventually gets transformed into long-term memory, that requires the hippocampus. So this is the kind of work that allows us to understand how the brain is and where the brain is storing kinds of information. So the hippocampus is a very interesting structure. Can you see it in this little video? Yeah? It's that red region. So that's where your hippocampus is. And so it's believed now that the hippocampus is involved in storing short-term information, and then the cortex is involved in storing the longer-term stuff. There's some very interesting things nowadays we do with the hippocampus. For example, the hippocampus has the property of storing information about place. So, for example, if you were to record from cells in my hippocampus, some cells, let's say it's the red cell over here in the corner, yeah, this cell over here, would respond if I'm standing here, but some other cell will respond if I'm standing over here. And another cell will respond if I'm over here. So if you record from enough cells, you can reconstruct the location of the human or the animal simply by recording these cells, which have sort of memorized the place in which they should be active. They are place cells. They recognize specific places. If you have a linear track, which is that S-shaped curve you see there, then you have a series of place cells that get active as the rat, in this case, runs along this linear maze. So the hippocampus is remembering many things, including the location of the animal. Yeah? And the cells are active when it's in a particular location. And I have to wrap up. So the question I want to bring is to try and tie these ideas together. How do you relate the synaptic level of activity to this circuit level of activity? In recent experiments, it's become possible not just to record from these cells, but also to stimulate the cells. And it turns out that if a cell is previously quiet, yeah, it's not interested in this place, but you trigger its activity right here, right? You trigger it a few times. That's the bottom few traces there. You trigger it over here, and now you go away, and you come back. That cell now remembers, oh, I was active over here. And so now that cell encodes that location. In other words, you have synthesized a memory. You have caused this cell to remember this location, and this has happened through associations. It's happened because the cell was active at the same time as input was coming from visual and other input. other senses to say that this is this location. So you're strengthening synapses for this cell in this location, and now it remembers this location. Yeah? And there are other lots of cool experiments. Here's another one which essentially says the same thing. You trigger place cell activity at a certain location, and now that becomes the location that the cell remembers. So these are some examples then of how you go from the very basic molecular level events through cellular events to kinds of memory that Pavlov discovered, and now we think about in terms of how the hippocampus works. So I'm going to leave you with this thought. You know, we now have many, many clues for memory. We know a lot about it, and yet there's so much still we need to know. We can, for example, we can erase memories by turning off the cells that are active in, say, a given location. You can just shut them off, chemically or nowadays optically. You can just shut them off, and the animal will forget that it was ever there. But if you let them come back again, it will remember. You can read out the location. You can make fake memories. Yeah? You can get cells to be active even though they would not normally have been, and then you can cause associations that never existed in the animal's real experience. Yeah? And as there's going to be a discussion session later, you can actually, there's some kind of relationship here between how neural networks work, because neural networks also rely on the connections between cells, sort of neural network nodes, to describe information. But we're still not sure how the brain does it. So there's a huge amount of work for all of you to do. Okay, so to summarize, I went down various scales of how the brain works, from the whole brain to cells to parts of the dendrite to synapses. I discussed how you can store information in chemistry. And then I discussed two examples. I discussed Pavlovian conditioning, and I discussed place cells as a way to show how these kinds of activities are helpful for storing information. Yeah? And I leave you with this thought. You have 10 to the 15 synapses in there. Use it. And if you really want to use it, do neuroscience. It's a plug for it. Thank you.
Thank you, Mr. US Bhalla, for an amazing talk. The floor is now open for questions. So, that guy over there at the back.
Good morning.
Good morning.
I had a question about hippocampuses.
Okay.
There is a rule in UK that if you want to be a cab driver,
Ah, yes. Yes.
so you should know the streets of UK.
Yes.
So is there a difference between hippocampus of a cab driver and...
Yes, so obviously you know the answer because you're asking the question. So it turns out that, like many things, if you use it, it gets strengthened in some way. And what this study, which he's referring to, showed was that these cab drivers in London, and by the way, unlike cab drivers elsewhere, these guys actually know where they're going. Yeah? They have to be able to find the location just by saying, oh, this pub name in some, just give the pub name, they should remember where it is. I mean, nowadays everybody uses Google Maps. These guys had it all in their hippocampus. And it turned out that the hippocampus showed some significant differences from you and me. Yeah. Wave your hand so I can see who you are. Okay.
Hi.
Yeah.
Thank you. Great speech. Two-part question, part
Yeah.
of the same thing. Is there a difference between Pavlov's conditioning and operant conditioning? Both are
Yes.
reward and all of that.
There's a difference.
There
yes.
is?
Yes.
Okay. And the second part is, you had mentioned that Pavlov's conditioning can be used in learning. Like, for example, every time I go to YouTube, instead of maybe learning how to play an instrument, I end up watching a cat video or something like that. So is there a way that, has this not been really explored fully, where you go to, you know, a certain area, and instead of doing something that benefits you, you ultimately end up doing something that does not really benefit you? So why isn't this being explored?
Oh, it's been explored. I mean, what you're talking about is reward circuitry. Yeah. So reward circuitry is another whole set of things. It is very closely linked to learning. In fact, there's a kind of neural network and real learning called reinforcement learning, right? What is it that gives you the sensation of feeling good, right? So maybe cats do it for you, right? But the point is that reward and learning are very closely linked. They have to be, right? for you to survive, you have to associate certain things with reward and certain things with punishment, right? But there's a lot of work being done on that, and that's another whole topic. So maybe you can talk later.
Hi, nice talk. My question is, we are born with 1 billion neurons or whatever. When we are
A lot more, 10 to the
lot.
11.
Okay, 10 to the power of 11 neurons.
Yes, 84 billion roughly, yeah.
But neurons don't make intelligence. It's far more complex.
No,
My question is...
Well, it depends. Okay, let's not get into semantics. Make is a loaded word.
Don't equate
Anyway,
to.
okay.
Don't equate to
Fine.
necessarily.
If you had a pile of neurons in a dish, it's not intelligent. Agreed.
Right.
Yeah.
Even within a human. My question is intelligence a variable or a constant?
Ooh. Okay, so there's many ways to define intelligence. Some of them have been very discredited. For example, IQ tests are discredited and very dangerous because they have led to all sorts of terrible racial and other kinds of prejudices and misconceptions. As with most computational neuroscientists, I would rather lean towards Alan Turing's definition, which has to do with the idea that if you are in conversation with an entity, human or otherwise, and at the end of an arbitrarily long conversation, you, as an intelligent human being, are not able to distinguish that entity from another intelligent human being, then it is intelligent no matter what it is. So that is the definition that I like, right? But certainly a pile of neurons in a dish, or as you point out, in somebody's head, do not necessarily equate to intelligence.
Anyone from the back side? Yeah.
Lots back there.
That girl.
Yeah, let's have this speaker.
Hello, we had a very great discussion. At last you told us that just in case we want to make new memories or we want to forget something, we have to destroy or create some cells. But
No, you don't destroy or create cells. No, you don't have enough cells to do that.
But how
The same
can,
cells, the synapses get weaker or stronger.
But how can we do that?
Well, it happens all the time without your particular effort, right?
Yeah.
The synapses, these are the contacts between cells. Because of when you have associations, these synapses get highly activated, and that triggers these chemical feedback loops. And that stores information.
Okay, thank you.
Yeah, so in other words, the moral to that story is that if you want to remember something, revise it. Repeated association is good for